feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Hi, and welcome to the show. This is the National University of Singapore, Yong Lu Lin School of Medicine's Healthy Longevity webinar. Thanks for joining us again. And please, I want to remind you to use the Q&A function to send questions tonight. We have Alaattin Kaya, who will be a really interesting discussion, I'm sure. Before we get to him, though, we have a short presentation by Yao Rei Hyun, who's an intern at the NUHS Center for Healthy Longevity. And the talk will be investigation of the effects of senescent exosomes on human bone marrow-derived mesenchymal stem cells.
Thank you for the kind introduction. Good evening, everyone. As mentioned, I'm Rayhan, and today I'm very honored to present our study on the investigation of the effects of senescent exosomes on human bone marrow-derived mesenchymal stem cells, or BMHMSC. Aging is described as the progressive decline of bodily functions, as well as a higher susceptibility to life-threatening diseases. We are currently facing a global aging crisis due to increased life expectancies and declining birth rates. In Singapore alone, it is estimated that by 2030, a quarter of the population will be aged above 65 years old. As a small nation that heavily relies on its people as its key resource, this aging trend will severely affect us. Thus, this warrants an urgent need to gain a deeper understanding of aging in order to promote healthy longevity. This brings us to one of aging's most studied topics, known as cellular senescence. Senescence often occurs in cells when they are exposed to damaging stimuli, either naturally in an aging body or artificially through drug intervention. In this state, cells undergo permanent cell cycle arrest, ceasing cell division and proliferation while remaining metabolically active. Senescent cells also secrete a unique composition of proteins defined as the senescent-associated secretory phenotype secretome, or SASP secretome. This SASP secretome differs from a typical secretome produced by healthy cells, and can induce senescence in neighboring cells through a process called paracrine senescence. As these senescent cells accumulate in the body, most often in the elderly, they consequently impair the function and regeneration of surrounding tissues, contributing to age-related diseases. To further explore the secretome, it comprises soluble proteins and insoluble extracellular vesicles, or EV. Among these EVs are exosomes, or exo, which facilitate cell-to-cell interactions by transporting cargoes such as proteins, lipids, and RNA. However, emerging evidence suggests that senescent exosomes within the SASP secretome are key mediators of paracrine senescence. Therefore, this study seeks to explore the paracrine effects of senescent exosomes on healthy cells. In our methodology, we utilize BMHMSC to replicate aging conditions due to their well-studied background and accessibility from bone marrow extractions. We cultured and split them into two different groups, a healthy non-senescent control and a drug-induced senescent control. The secretome of each group was then collected and processed by ultracentrifugation to isolate the respective exosomes. From there, we characterized the exosome surface marker profile from respective groups via flow cytometry to better understand and identify any differences between them. The paracrine senescence effects of the exosomes were also investigated by exposing them to healthy BMHMSC before evaluating any cell count changes via immunofluorescence staining and microscopy. For flow cytometric characterization, we analyzed the expression of several different surface markers of exosome and stem cell origins. we consistently observed a trend of higher marker expression in senescent samples compared to non-senescent ones. Additionally, CD44 stood out as the only one with a statistically significant difference among all the other markers. This increased expression of CD44 in senescent samples suggests a possible relation to senescence. Moving on to our exosome exposure results, after 24 hours of exposure with the respective exosomes and controls, we quantified the cell populations over 3 days. We observed similar increasing trends between the non-senescent exosomes and unexposed or untreated negative control. On the other hand, we observed comparable fluctuating trends between the senescent exosomes and drug-induced positive control. This drastic decrease in cell count could be attributed to factors such as increased cell death or decreased proliferation rate, which correlates to senescence. To conclude, the demonstrated link between senescence and CD44, a well-known cancer stem cell marker, requires further investigation to verify its potential as an aging biomarker. Moreover, the ability of exosomes to induce senescence in healthy cells indicates their potential as senescence biomarkers as well. Moving forward, we plan to expand this study and further investigate the paracrine senescence or BMHMSC-derived senescent exosomes in other in vitro and in vivo applications. Through this, we aim to advance translational aging medicine with the goal of developing interventions to impede aging and treat age-related diseases. And with that, I've come to the end of my presentation. I would like to acknowledge the HLTRP team consisting of the following members for their guidance. Thank you for your attention.
Thanks a lot. More information about senescence, and the field just gets hotter and hotter. We're going to change course a little bit, though, and talk to Alaattin Kaya, who's on the faculty at Virginia Commonwealth. He's a geneticist and a biochemist who focuses on various aspects of aging biology, from evolutionary molecular genetics and biochemical viewpoints. He studies the interaction between genes and environmental factors in aging patterns across organisms. And I think that I could go on about this. You have a lot of other interesting projects as well. But I think I'll just focus on the one today, which is multi-omic and multimodal approaches for target discovery against aging and age-related diseases. Alaattin, thanks for joining us.
Thank you for the invitation, Brian, and I really appreciate that. This is an exciting opportunity to present here. So I would like to start my talk then. So today we are going to talk about how my lab applied these multimodal and multi-omic approaches to understand the biology of aging and some diseases. So this is the outline. My lab has been already almost 4 years since open. Most of the projects are still ongoing. Some of the data I will show you today is published. Some of them is unpublished. So we will talk about 3 topics. But before we start, I want to introduce you some concept that different models has been used in the field. You know, each lab used different models, different approaches. I think the most famous one here is the yeast, C. elegans, fly, are the invertebrate models. There are translational perspectives of aging studies, mainly use mice model, and then of course we use some human samples and human cell lines to understand the particular question related to aging. Each model, of course, gives you a different aspect that depending on what question you ask. So although one thing is I need to highlight here, the evolutionary distance is really huge between these organisms. We know that it is aging conserved, right? So in the field, I realized that one thing is missing, that there's not much attention, really, what is the conserved regulators of aging across these model organisms, right? So basically, we use these different models in my lab and ask the same question. So what are the conserved regulators of aging across different species? So I will start in the first project that we started 3 years ago. This is a network. Basically, you can see that different gene names. The black ones here are the essential genes here, and randomly chosen almost 50 essential genes. And then the red ones here that showing that the genes that are characterized or involved in aging process in different organisms. The highlight or take-home message here is that you can see that these essential genes directly interacting many regulators of aging. This is very striking, actually. And we get this data, and then we ask that, is there anybody work really on how essential genes are regulating aging? But the problem is that we found conserved essential genes are really understudied in experimental aging research. And then we wanted to jump into this field, and we hypothesized that these essential genes are highly conserved from yeast to human. And we think that it might be largely untapped source of the pro-longevity genes. But the problem is that most of the genetic screen examine loss of function, like siRNA or gene knockout or decreased expression through the reverse genetics. And essential genes have often been overlooked as a potential modulator of aging process because we cannot really manipulate them in the genome, right? So what we did is we used yeast, and we decided to overexpress these essential genes. We used the replicative lifespan assay in the yeast, So this is very famous in the field. And briefly, you get the virgin cells every cycle 1.5 hours. You keep that virgin cells and count the budding, and basically how many daughter cells coming from this mother. And then you plot on the right, you can see the red is living longer than purple, living longer than green. So this is a well-established yeast aging model. We call it replicative aging model. So we use this approach. And then in yeast, there is 856 essential genes conserved in worm, in human. So this is really striking, actually. An essential gene is evolutionarily selected to be highly conserved, and they are generally escaped from the mutational process. And there is a recent paper showing that out of these 856, 621 of them can be exchanged with the human orthologs. It means that functionally is conserved these genes. So this is a great data. And we did preliminary screen. When we overexpress one copy, we increase each essential gene, and we see a striking result, actually. On the left here, you see that these 856 essential gene, red shows the decreased expression, green shows the increased expression, gray shows no changes. And then on the right is non-essential gene. We tested 50 of them. And essential genes, what we find, almost 200 of them, which is 23% of them actually, increase the lifespan 13 to 76%, which is very striking. And this was quite shocking actually to us, but it was a very exciting data, right? So another thing here is that seems like expression of even the non-essential genes has a higher rate of almost 8% is lifespan increase compared to these knockout library screens in worm and yeast, in which they characterize almost only 4% of the knockout genes increased lifespan. Seems like gene expression might be a really potential to identify these conserved regulators of aging. And these genes are in different cellular processes, you can see. So there are DNA replication genes, ribosome biogenesis, spliceosome. So overall, there are different processes maintained by these essential gene regulators. And at the end, they somehow regulate the lifespan. So one example is these two genes that has never been worked in the aging field. This is the wild type, this is the GDA1, and wild type and TIF31. There are two different processes, these genes. You can see a huge, significant lifespan extension in this. This is just an example, right? So we did some preliminary characterization, for example, and you can see that when we overexpress one copy, putting one copy of this gene into the genome, you can see that we increase the overall dosage almost twofold. There are, for example, here, this is the PCA plot, wild type is here, LSM5, PRP, TIF, and UTP. The data showing that each of them induce different transcription network or transcription regulation, distinct to each other. Basically, what we can learn from here is that the data indicates twofold increase in mRNA level of essential genes is sufficient to changes in the cellular processes, and basically through the transcriptional changes. In addition, our data suggests overexpression of essential genes might extend lifespan through different mechanisms. That's what we are proposing here. And On the next stage, of course, is this conserved, right? So we are talking about conservation. We choose another distinct organism, C. elegans. Basically, now currently we are trying to generate transgenic animals that those 200 yeast essential longevity genes we are overexpressing in worm. We try to approach one, a classical injection. Another method is we do this sgRNA feeding. It is recently published by the Risto group, and basically it's working great. So we do this worm with the that Cas9, and then when we put the sgRNA feeding targeting the promoter region of each conserved essential genes, we overexpress, induce the expression of those essential genes. The next stage, we will choose some strong candidate, and we will hopefully get some mice models overexpressing these conserved genes and test the lifespan effect. So we use this worm bot, Kaberlin group published, and we have in the lab. We should through the Nathan Schack Center, and it's provided by Kaberlin group. You can see that it's an amazing tool that you can easily characterize the lifespan of any model, your worm model, how it affects the lifespan here, right? So we have this technology in the lab, and we apply. So these are the 4 different genes you can see in the yeast. These are the essential genes. Red is significantly increasing lifespan. Again, the question, is it working in worm? Is it conserved? We choose the same gene orthologs here. Out of 4 randomly chosen essential genes, 3 of them significantly extend the lifespan. The data is very robust. Each data consists at least 150 worms and 3 repeats. This is the combination. So this is very striking, actually. If you think that you choose randomly 4 genes and then putting in the yeast, it extend lifespan. Out of these 4, extend worm lifespan. So that's very striking for us, and we are working on this data. So one example is this, how the mechanism, right? So it seems like if you look at the young and old comparison, these are the young overexpressing compared to wild type. There is some genetic expression difference, but seems like in the old yeast, the gene expression changes is very significant. So we hypothesize that probably the regulatory gene expression changes in the late life influence lifespan in yeast strain overexpressing these essential longevity genes. And probably in the late life, there is some dysregulation of these essential genes. And when we put X-ray copy, we compensate it simply. Okay, in conclusion from this part, basically increasing the expression level of conserved essential genes, we found 23% of them resulted in significantly increased lifespan. This is greater than the almost 4% genes found to affect lifespan upon deletion or decrease expression. Along with the lifespan data from worm, I would say that this is a very potentially significant project. Hopefully, we will identify many unknown genes that has never been characterized in the aging field. Okay, the second project we focus on is we use this robust model, wild yeast isolates. Why? Because in the aging field, I realize another problem is we stuck with the laboratory adapted model organism from yeast to mice. So when I open my lab, I bring these yeast model collected all around the world. They are all budding yeast species. And the striking thing we published previously, they have huge genetic diversity, although the same strains of the same species. And this genetic diversity, of course, brings a really huge phenotype in the lifespan. You can see that we analyzed the lifespan in 3 different conditions. This is the high glucose, this is the glycerol, which activates the mitochondria and known to extend lifespan in the lab, and this is the CR. What we saw here is that there is a lifespan diversity under each condition. Even in the CR, so we see a lifespan extension in some strain, but we see also decreased lifespan extension in some other strain. This is showing that genetic background is really important for any intervention we are testing. So this is a great model. How can we identify this phenotype of lifespan diversity, right? So we use some of the endophenotypes like transcriptome, metabolome, and proteome to understand how this genotype is associated with this genetic lifespan diversity, right? We just put actually one preprint recently. For example, this is the CR-mediated lifespan. We find that the strain positively responding CR has a huge gene regulatory network mediated by mainly GCN4. Upregulation of GCN4 upregulate stress response genes and mitochondrial respiratory genes. And in the end, when we delete the mitochondrial genome or repress the mitochondrial respiration, you can see strains decrease the lifespan under CR condition. And overexpression of GCN is alone enough to induce lifespan extension. So it seems that something like mito-UPR is effective. That strain can upregulate this mito-UPR under CR, positively respond to CR. And we find some metabolites actually nicely correlate with the positively responding or negatively responding under CR. But one striking thing is that we find N-formyl methionine. So this is the methionine, form of methionine. It starts the mitochondrial translation initiation positively regulate with the CR response. It means that higher N-formyl methionine strains respond robustly to CR. So that's a very striking. It has never been associated, and we are very excited about this data, and we are doing some extra work on this. How about the regular lifespan under normal condition? We found that most of the strains completely decrease all genes involved in branching amino acid. All these rat genes are decreased expression in the long-living strains. So they decrease the branch chain amino acid synthesis. Another striking thing is we find the tryptophan synthesis. So we find tryptophan increase in the cells with long-living cells because we find that this BNA2 is decreased in the expression. So this is the correlation. This is basically the count of the tryptophan, and this is the median replicative lifespan. Really nice positive correlation. Tryptophan increased because the first gene, you might remember that BNA, which catabolized the tryptophan to kynurenine, is decreased expression. So, and one of the intermediate metabolites you expect here decrease, right? And it decreased. So this is very striking, actually. So why such a pathway is tightly regulated in the long-living strains, right? So tryptophan is a very interesting amino acid. In higher eukaryotes, it's come only through the diet. And BNA2 is the first enzyme catabolizing this tryptophan to kynurenine. In mammalians, there is IDO1, IDO2, TDO, three genes. And in yeast, it can be synthesized, and it can be gotten by diet. And there is a recent exciting in the aging field, this serotonin-melatonin pathway, which is actually derived from the tryptophan metabolism as well. So there is a connection here. So we did some analysis. In the yeast, you can see that when yeast age, this BNA2 expression is increasing huge. And then same in the worm. The worm that get older, BNA ortholog, IDO is increased expression. But what we find is decreased BNA2 expression is actually beneficial. So we test in the yeast. So you can see blue is wild type, red is genetic model of hexakinase 2 CR, extend lifespan. When we decrease, completely inhibit the BNA, the lifespan effect is lost. More interesting thing is that the CR cannot respond anymore. So it means that this BNA2 activity is required for the CR to extend the lifespan. But that's kind of interesting. And we test on the worm. Here is the siRNA. This is the N2. And N2 on TDO, which is the BNA2 orthologine. It extends lifespan when we decrease the expression. And E2 is the CR, and it extends lifespan. But when we combine TDO and ER, we see striking lifespan extension. It's a combination of both intervention. It means that it extends lifespan independent of the CR. But the thing is that we see same in yeast. When we completely knock out the TDO, all this lifespan effect is lost. It means that the minimum level of BNA or TDO expression is required for lifespan extension, especially for CR-mediated lifespan extension, right? So then we said, is this conserved? We get the mouse model. So in the C57 Black 6 model, there is this IDO1 and IDO2 on the same chromosome. We got the IDO1 knockout. It's published previously by deleting some of the exon. We got the IDO2 knockout, and then we got the double knockout of the C. But the exciting thing, we also generated this model in UMHAT model. So we got two different strain background, because I told you strain background is very important for any intervention. And then we got UMHAT3, all these three knockouts. and we start lifespan analysis. Yeah. Currently, our chronological lifespan analysis is ongoing. Umhat is almost 5 months old, the colony, and then C56 is almost like 9, 10 months old. And so far, we don't see any phenotype. They are living healthy and happy. And we are doing some downstream analysis. The one striking thing you can see here is that although the blocking these genes decrease this kynurenine pathway, the exciting thing is that the NAD level, which is the tryptophan catabolism is the major pathway. We don't see any changes in the NAD level, actually, in these mice. This is analysis coming from the C56, 57, black 6. So there should be some compensatory pathways here. So first we get this C57 black 6 4-month. We did some brain and liver transcriptomic analysis, right? So here is the data coming. There is a beautiful evolution here. What we see is both, don't worry about the numbers, but you can see that there is a there is a sex and tissue-specific effect. For example, in the IDO2 knockout, this is the male. You can see there is a huge gene expression changes in the male, but not in the female, versus in IDO1 knockout, male versus female. So the overall data showing that there is a tissue-specific effect of each deletion, and there is a sex effect. So that's kind of exciting for us. And same thing for the downregulation genes. Brain and liver seems like IDO2 mostly affecting brain, IDO1 mostly affecting liver, and then there's a sex-specific effect on that. So we use this transcriptome data. There is a manuscript in preparation by Gladyshev group. So we developed this transcriptome-based aging clock, and what we find is actually based on this transcriptome, the biological aging in IDO2 and IDO2 double knockout is decreased, meaning that there should be a really expected lifespan increase, these two in the female. And in the male, we see the very striking effect in all background. So that's kind of really exciting. It means that our lifespan, let's say when we finish, we might see a really significant lifespan differences. It indicates that decreased tryptophan catabolism might induce lifespan conserved from yeast to mice. And we see also relative biological age decrease, similar pattern. And of course, sample number is low, and this is the 4 months old. We think that when we get older age and more sample number, we will really get a significant differences in the lifespan. Seems like this is an exciting field that tryptophan catabolism, decreased tryptophan catabolism might induce lifespan extension. So, and we have some mechanistic insight. We think that there is related to gut microbiota. Why NAD level is not changing? Because gut microbiota is colonization is different. NA, some of the precursor of NAD is supplied through the gut microbiota in this mice model, and there is a lifespan increase probably due to this. We are investigating this. And basically this project is ongoing. Now we are analyzing UMHAT to see the strain background effect. It's very exciting. We have a same, strain background in C57 and UMHAT mouse. Hopefully, we will see more striking effect in this mouse, genetically diverse mouse background. Of course, we will analyze strain, sex, specific effect, mechanistic understanding, and of course, recently there is a huge attention that this tryptophan catabolism and serotonin is very important for regulator of neurodegenerative, especially Alzheimer and Parkinson. So we are also have a project in this direction as well. Finally, the last project I want to show you, also using this interspecies and omics data so we use this disease similarity approach. We get the data from different databases, and then we apply some machine learning approaches, and we are trying to identify some drug that can be effective in some neurodegenerative diseases. Here is an example. We get different diseases, which is known to basically the increased risk of onset Alzheimer's disease development. Like Down syndrome is known, Niemann-Pick disease, this MCP or I never say the name, diseases. But these are the diseases the patient has a basically higher increased risk of developing Alzheimer's disease. Two things I want to highlight here is Niemann-Pick type C disease, which is very rare disease and barely work in the aging field. So I'm very excited to bring this model into the field. So we use this Niemann-Pick type disease and Alzheimer's disease comparison in this project. So what is Niemann-Pick disease? This is basically two gene NPC1 and NPC2 mutation cause the cholesterol inability of cholesterol internalize in vacuole, and basically it causes neurological diseases, and it's very rare. Basically, you can see this estimated prevalence is from 1 in 2.9 million, and that's because very rare and not much people working on this. There is no known cure. There is no known drug against this. But the thing is that this Niemann-Pick disease, also known, if you Google it, it's known as a childhood Alzheimer, because the patient don't live more than 15, 16 years, and they start developing all these hallmarks when they are 12, 13. So it's very similar to Alzheimer's disease, right? So we got this disease, and we got unfortunately, we have no human samples and no omics data from human samples, but we get a really nice mouse model which recapitulates the human NPC very nicely. You can see NPC mice start decreasing the weight. They die very early. They generally live around 120. This is a mixed sex. They have basically visible tremor is increased. These are all other phenotypic functions showing that motor function is lost. There is a basically neurodegenerative defects in motor neurons start very early. So these are exactly the same phenotypes of the human diseases, right? So then this is a nice phenotype data. Then we compare this NPC mouse versus one of the most commonly used Alzheimer mouse model. Here you can see that APPPS, which is a double transgenic mouse. We found actually in the male and female some overlapping genes, but also there are huge transcriptional diversity between these two mice models. But I should highlight that there is none of the mice model is the best recapitulate Alzheimer's disease. And here it is. I want to point out that when we compare the transcriptome data in human brain samples or organoid samples on the top, you can see here, and then on the bottom, downregulated, upregulated, I want to show that our NPC mice actually has more common genes in human Alzheimer samples than the most commonly used APPPS mouse model. This is very striking. It means that this mouse model can be the better model to work on the Alzheimer, right? So we find, but most exciting, some common genes actually commonly overexpressed or upregulated or downregulated in all samples, in AD, APPS mice, NPC mice, or brain samples. And one of the genes we are very excited is TPD52. It's overexpressed in human brain AD, mouse brain AD, and NPC mouse. So this is one example. We use ML-based approaches. We characterize some molecular genes, like mRNAs can be targeted to down-regulate the expression. But we also find a really unknown drug, zeledronic acid, strongly bind this protein and inhibit function in vitro. So now we are testing this the molecule if it is effective against Alzheimer's. So this is showing that how different species can be used against these human diseases, right? And we are very excited about this NPC mice model. And the last couple data, I'm almost out of time. We use the same transcriptome clock in the brain. You can see that relative expected lifespan as consistent our data in NPC mice, male and female, significantly decrease, very significant. And then the biological aging rate is really increased. So the brain age very faster. So this is a nice model, short-lived mice model, only lives 120 days. And we have a really great accelerated brain aging here. You know, the brain age faster. They accumulate all these hallmarks of Alzheimer similarly that we see in the human in this mice model. And we are very excited about putting this model out, and we are preparing the manuscript currently. Soon, hopefully, we'll put it out. Basically, there are conclusions here. Basically, different models, especially NPC mice model, can be a better model to understand the human AD pathology and genetic regulators. Machine learning-based strategies can be applied in this mice model to identify drug. An overall cross-species multi-interdisciplinary approach will be the best effective strategy against the age-associated diseases. With that, I would like to thank our collaborators from VCU and from University of Washington, especially Caberline Group, and from Columbia, Vijay Yadav, and from Harvard, Vadim Gladchev and Alexander Tishinkowski. And my lab members Naji involved in the essential gene project, Vikas involved in this disease similarity approach, and Rohil and Murtaza involved in this mice model of tryptophan. And basically our funding source I appreciate. Without them, we couldn't do any of these projects. And I'm happy to get any question if you have.
Thanks. That's a great talk. A lot of questions, but before I get to them, I want to apologize to the audience because I don't know if you remember. I was probably not. I was doing a show a few months ago in a room where the lights keep flickering, so I came back to that room, and so I turned the lights on really early to make sure they weren't flickering this time. And as soon as the show started, one of them started flickering. But I really care about my audience. So I was precariously climbing on this fragile chair to unscrew that light and thought I solved the problem. And now the second light is flickering. So if I keep unscrewing lights, we're going to be in the dark. So I guess a flickering Brian is better than a dark Brian. But sorry if that bothers people. Um, so lots of questions. Maybe we'll start from the back and work backwards to yeast. The Niemann-Pick syndrome mice. Do they have phenotypes outside of the brain, or other? Is aging accelerated in other tissues as well?
Yeah, so currently we analyze on the brain. Yes, I mean, within 90 days, 3 months, these mice even cannot eat the food. They cannot reach the food. We had to put the gel food. Their muscles are degenerate very quickly. And the cognitive decline, yes, there is a cognitive decline, very obvious in the third month. This is very similar to human, actually. The human patients start when they are 12. They start all this cognitive decline, muscle degeneration. That's why Niemann-Pick type C called the childhood Alzheimer's. And I'm surprised that nobody really utilized this model, bringing aging, accelerated aging, and then the Alzheimer model. And what we find is the gene expression similarities really robust in this mice model compared to human frontal cortex than any known mice Alzheimer model. And of course, this is a very striking, and we find these common genes as well. But we're also looking on the human NPC commonalities, removing all these mice model, and then looking at some new regulators we find. But one thing we are excited is this TPD52 and MIAT gene. That recently MIAT has been a little bit associated with the Alzheimer, but not much known. So we identify many hop genes that can be actually important regulator of Alzheimer progression, and we are currently working on that.
A number of the geroprotector drugs, particularly rapamycin, but others as well, have been suggested to have protective effects in neurodegeneration. I wonder if you've tested any of those in this mouse model.
Okay, so this is the thing. We already have a huge mouse colony UMHAT and then C57 black for the tryptophan project and this NPC project. So the cost of the mice, so I'm currently all applying grants, but unfortunately I'm not successful currently, although I get some grant for the other projects. And I'm trying to find more money to apply. Actually, this is the first thing we want to apply caloric restriction. And then we want to apply rapamycin on this mice model because it's a short-lived, and we want to see if rapamycin and then calorie restriction can elevate some of the phenotypes. We will start. We have the mouse here. We have breeding colonies already ongoing. This is a very exciting mice model, actually, and I hopefully we are preparing manuscript. Once we publish, people will pay more attention to this mice model and will utilize in different perspective of the aging. Yeah.
Well, I think it'll be fun to see what emerges. And also to compare it with some of the other progeria models too, and see what there's commonality.
Yeah.
I think that the thinking on these progeria models is that they're segmental. That different genetic mutations accelerate different aspects of aging, maybe in different tissues. And so this seems like there hasn't been a good one for the brain, really, because the Hutchinson-Gilford mice don't express that splice variant in the brain, and so they don't get any phenotype of the brain. So this could be a really good model to look at, I think.
Exactly. And another thing is this doesn't accumulate many mutations. The lipid lysosomal dysfunction, which is actually recently suggested as a hallmark of Alzheimer and aging, right? So this is a very new perspective, not much basic. This is not a hypermutated strain, So that's very interesting to see if other organs also aging faster. That's what we are doing now, and basically that's a very I assume that this is more physiologically better model to characterize brain aging than those progeria. And I didn't know that progeria. I never worked, but this is I'm assuming that accelerated brain aging is a really nice model here.
Let's spend a few minutes on yeast because I don't get to talk about yeast that much, so
Yeah, yeah,
it's me excited. So just for our audience who's probably not thinking about yeast genes a lot,
yeah,
the there's always been a over reliance on studying non-essential genes because the much of the traditional approaches to yeast involve knocking out particular genes, and even yeast can be haploid or diploid, so you can knock the gene out in a haploid and examine the effect of the gene. You can knock out one copy in a diploid, but the phenotypes are not seen as much, as at least that's what has been reported. So this idea of overexpressing them, I think, is really intriguing. I guess it's not surprising that essential genes are more likely to regulate replicative aging, but the frequency of genes that you hit are so high. It's really striking.
yeah. This is a very exciting project, actually. When I opened the lab, this is the first project we start. So in my lab, I try to go a little bit outside of the question that not many people working in the field, right? So basically everybody work on the lab models. I try to avoid that, bringing different strain background, different models. Also, one thing that when you look siRNA screen done in these C. elegans, knockout screen done in the that those are really informative and very useful, right? We've characterized many, but everybody avoid this overexpression. First of all, I don't know why. And second, everybody avoid essential genes. Our network shows that actually most of the essential genes involved in a large complexes where other non-longevity genes are involved. So I said, how about this? We increase the one copy of the essential genes and see what happens, right? This is a simple question. And in nature, actually, we don't see too much gene loss. Look at the cancer, look at through the evolution, gene loss is very rare. Most of the time we see gene duplication. So this is a question that gene duplication is beneficial in the evolution. Fitness increase, probably other stresses. And so based on this, I said, let's put one more copy of the essential genes because it's conserved through the evolution. And we see this striking 23% of the lifespan increase. When I did a pilot screen for the 100 and we get this number, I talked to Matt Keberline. He said, this is something wrong. Let's repeat it. And we got this Nathan Shack.
That's always what Matt says. So that's
yeah. So we got this Nathan Shack pilot award and same 100 strain tested. In my hand, they were like around 25%. When they tested, they hit around 22%. So it's very striking. Then he was very excited, right? We start this project. Of course, yeast is nowadays, I really love this model. I work with mice now, C. elegans. I will never leave this model. It's very, you can do lots of genetic screens, lots of innovative approaches you can test in the yeast and transfer to the mouse, like this essential gene. Many people avoid working with yeast. Even the getting grant is really hard nowadays. Using
it's
yeast.
really unfortunate. As someone who worked with yeast, there's a lot more we can still learn in that system. But
Exactly.
right now it's being dominated by this concept of translating things and not making the basic discoveries that are going to set the stage for future translation. It's kind of penny wise and pound foolish, I think.
Exactly. People asking me why I still keep working with yeast. I tell them, maybe in the future I will drop mice, but I will never drop the yeast. So this is a very exciting field, and I will basically keep working on them. So
I'm going to hold you to that. So I'm going to come back in 5 years and check and make sure. So
okay.
the wild yeast are quite interesting. We had that idea a long, long time ago to look at some wild yeast isolates. But back in the day, the isolates we could get were really hard to work with. They're clumpy. It's hard to get the daughter cell from the mother, and we couldn't find enough strains to really get much information out of. But another feature of these wild yeasts are as far as I understand, they're often aneuploid too. So it might be worth just looking at copy number of all the different genes
Yeah.
and see if copy number of specific genes correlates to lifespan extension.
Yeah, I mean, we look at and some aspect going. Actually, we have almost 1,500 wild isolates. The genome is recently published in Nature 1011. So yeah, many of them is clumpy. It's very hard to dissect. We choose the best 1,800 of them to dissect, and out of these 100, we have only 6 of them is aneuploid. So and we should look more aneuploid strains. This is a nice idea. But we did some GWAS. And we find some association with some of the SMPs. We haven't particularly looked gene, even though there is no aneuploidy, there is some gene duplication, and there is some unknown genes in these strains that has not evolved to another strains. We haven't looked at those, but that's a really nice idea.
Yeah, I think there's probably a lot of information in there. Let's get to the IDEO 1 and 2. First of all, that looks like a gene duplication itself. Those two genes are right next to each other. So what's the difference between IDEO 1 and IDEO 2? Do they have different activities?
Yeah, seems like IDO1 has higher tryptophan catabolic activity and mostly overexpressed in liver and immune cells. IDO2 is mostly neuronal and then brain. And basically it's a gene duplication, but then throughout the evolution they become tissue specific, and then the function little will change from each other. These are the known function. And what we show in the transcriptome also, there's a sex specific, like IDO2 more effective on the brain in male but not in female. So there is a sex specific evolution here as well. So this is very quite striking actually. And in the field recently, this serotonin melatonin is getting more attention. Plus, tryptophan is always coming out from many studies, including our studies. On this study, really early, tryptophan restriction increased the lifespan in rat. But other than this, nothing really known. But what we saw is decreased tryptophan catabolism. And George Sutpin from University of Arizona recently published a group, one of the intermediate is very effective to increase lifespan in mice and in C. elegans. So we find that actually the blocking, decreased, not complete inhibition of this tryptophan catabolism is somehow very effective to increase, seems like potentially increase lifespan in a
Is mTOR affected in those mice?
Sorry?
Is mTOR affected in those mice? I'm curious whether mTOR pathway is affected.
Yes, in some based on the sex dependent. I don't remember exactly which background and which sex, but we find some mTOR dependent pathways affected.
That's interesting.
And
Well, we'll look for some more data.
Yeah, the interesting thing now we have UMHET mice too. We have C57 black 6, we had UMHET. Now we are comparing strain effect, strain dependent, sex dependent, and tissues dependent effect of this lifespan. Is very exciting. And we will see what we will end up basically.
Okay, maybe what I'll do is bring Jianhua and ask some questions from the audience.
Thank you, Professor Alaattin, for your insightful talk. So here are some of the questions from our audience. So basically, the conserved genes that increase lifespan when overexpressed, why have evolution pressure not selected for more of this gene to be expressed? In other words, does overexpression of these genes confer a disadvantage? And there's an add-on to this question will be, do you manage to check for the fitness effects upon expressing these genes?
So this is a really nice question. I think there is so much evolution and biology, this essential gene regulating this essential gene expression. Many essential genes are highly abundant, first of all. And most of them, we try initially high copy expression, and we see all fitness decrease. So there is a take-home message and very important point here. We only duplicate one more copy. And as you see from transcriptome data, they only increase 2 to 3-fold expression. When we go over 5-fold, we see huge fitness decrease. They start growing because of the dosage toxicity. I think these essential genes are very important for the development. The increased expression might be interfere with the development. So that's why we are trying in the worm different combination of expression. This sgRNA-based overexpression model is really nice. We feed the worm during development. We feed the worm after development to see this development effect. So I think it's somehow related to developmental process, why these essential genes not being highly expressed. But in the cancer, what we realize is this: many of these essential genes duplicated in the cancer. So it means that there is some fitness advantage based on the context of the function. Yes, some of the gene that we find, even though 2-copy increased lifespan, they grow slower. We realize that. But most of them with 2-copy increase do not have any fitness effect in yeast. Now it's exciting to test all these questions in the worm. But you see that out of 4 randomly selected worm, these are the injection model, meaning that it's a genome integrated. They are expressed throughout the development and adult stage. We don't see any fitness decrease in this level of only 1-copy increase. But we will also now high throughput screen all these essential gene development versus adult stage, and probably we'll see some of them will decrease fitness. But seems like many of them will not affect the fitness at this level of dosage increase.
Just to jump in there, we were on the GCN4 pathway a long time ago for aging, and we never could find a way to overexpress GCN4 and get lifespan extension. So, I think so. You just did that with one extra copy, or how did you get that
Yeah,
phenotype?
you put ADH promoter. We realized the same
Yeah.
thing actually. When you put the strong promoter, cells are screw up. They decrease the growth. You cannot even. So we try different promoters, and you eliminate this UTR part. Just directly start from the transcription side, and you really put the weak and then the low copy plasmid. That's the problem. When you start hitting another thing interesting, GCN4, even the low copy expression in the row 0 strength that the mitochondrial respiration is eliminated, even the low copy, we couldn't get a really healthy cells. I don't know. I couldn't find any paper and on that. Yeah.
I don't know. Didn't know that either. I'm not sure what that means, but it's interesting.
Yeah, that's interesting. But you need to, when you overexpress GCN, you use the weak promoter and low copy plasmid.
Jianhua.
Yeah, next question. Is regarding the start of your presentation. In one of your in your second slide, you actually discuss about the network of essential genes and age-related genes. So how often, what is the reproducibility level of getting similar age-related genes when a random selections of essential genes were used?
So in that network, it's a simple network. Basically, we randomly choose 50 essential genes from different, when we say random, each gene has play role in different pathways, basically, not similar pathways, right? We throw into this program which generate network and essential gene. There are many papers showing that evolution conserved, and they are always involved in protein-protein interaction. So that's what we see in the network. Even though 50 gene, lots of connectors, right? Interactors. So there is a high chance. This is only 50 genes, actually. Choose another 50 genes. We did. You always hit some of the aging genes. Choose 100 genes. Then, of course, your level of hitting those aging genes increase. But there is some other group published a paper from Gary Rauken group. Initially did some essential gene knockdown in the adult stage, which is important for development, but not for the adult stage. They see the same thing. Those essential genes most interact with these known aging genes, and there is other computational based paper published from different groups. They are really involved in dense network of protein-protein interaction network, and you always have a high chance of hitting those one of the aging genes.
Yeah, thank you. And last question will be, how do you manage to produce all these RLS data? Are you working with microfluidic chips or by hand?
So the first 100 of them is done by both my lab in the tetrad microscope, and also done by Nathan Schack Center in the University of Washington, Seattle, also microscope. So one thing with the microfluidic, there are tons of microfluidic published, right? So we try to bring some of them into lab. Unfortunately, none of them work in our hand as it's published or advertised in the paper, So, and they are not high throughput. And basically now I train several undergrads and then graduate students. They can analyze up to 12 strain. Each of them has 30 individual mother cell. So this is a high throughput. At the same time, 360 cells we can analyze. Although replicative lifespan is very intense, time and manpower is really intense on this assay, but we analyze all of them through tetrad dissection microscope. Other than this 100, when we do this preliminary screen, of course, this is 856 of them. We didn't analyze 30 of them. I need to highlight here. We only screen 10 mother to each. This is the method. It's also applied by Brian Kennedy and Matt Kaberlein group, and they screen all these knockout. So this is the preliminary data based on 10 mother cell per strain, this essential overexpression. But now we have this 200 candidate. Now we are scaling up, getting each of them, repeating that data with a higher number of mother cells. And we are using this tetrad dissection microscope, not the microfluidic.
So that's music to my ears. Matt and I probably dissected more yeast cells than, at least at some point, we dissected more than anybody, I'm sure. We'll have to have a competition between our labs. I'm a little bit out of practice though, so I have an excuse already.
We should. I have a microscope at my home, small incubator. I dissect still by myself. I take some plates over there. I don't know how many, all these wild isolates, it was entire PhD, so I did. And then these essential genes, I did a lot of tetrad dissection, but I'm getting old now. My eyes are not as good as the old time. I can analyze 15 strain, 30 each, 415 individual, I was good at that. But when you get old, actually, this replicative lifespan analysis is really becoming harder than harder. So
that's what I did too. I just stuck the microscope in the back of the car and took it home on weekends.
Yes. Yeah, exactly. We did. We have 5 dissection microscope, and one of them is at my home. I do dissection time to time.
Let's come back to one other question I missed on the wild yeast, which I think are fascinating. You said there's decreased branched chain amino acid pathways in long-lived strains. Was that the correlation?
Which one? The decreased branch chain amino acid?
Yeah.
Yes, exactly. All those genes, if I can go back, but I need to share data. All these genes, ELV, BET1, BET2, very strikingly, all of them decrease their expression, like in all these long-living strains. And at the end, we see a decreased leucine level, for example, decreased valine level. And recently, there is a group from Princeton, I forget her name, Colin Murphy, actually published that BET1 or BET2 inhibition increase lifespan in worm 2. So it seems like our data is very consistent, like decreased branch chain amino acid is evolved in these long-living strains, so they synthesize less branch chain amino acid. And Dudley Lamming group has really lots of paper on this field. They decrease the restriction of these branch chain amino acids seen in the mice lifespan extension. Seems like a conserved.
Yeah, well, the reason I bring that up is that I was working on maple syrup urine disease, which is a branched chain amino acid disease, and the lab just kind of stumbled on the fact that a lot of the JR protectors that extend lifespan and control mice also reduce branched chain amino acid regulation, and so there may well be a connection there. We could talk forever, but I'm running out of time. So thanks for joining us, and I want to hear more about those mice in the future.
Of course, anytime. And if you have any ideas, we are happy to collaborate. Send the samples, send the data, and I appreciate this opportunity here. It was exciting, and thank you for the invitation.
Thanks a lot, and we'll talk in the future. I think there could be ways to work together.
Okay.
Thanks for joining us. I want to remind everyone to use the chat function and panelists and all attendees option to leave comments and feedback on the show. The latest work from one of our esteemed assistant professors, Vincenzo Sorrentino, has been published. It's a collaboration with Nestle Research in Switzerland and a number of universities around the world. And it shows a functional link between the natural alkaloid trigonelline and NAD metabolism and muscle health during the aging process. Basically, they discovered that trigonelline is an NAD precursor that can enhance muscle and mitochondrial function during aging. This is an endogenous metabolite, so it's another potential natural product that might affect the aging process. This was published in Nature Metabolism, and there's a link at the end of the show in the chat. The next show will be on April 4th, and we'll have Mitchell Lee, who's a founder of Aura Biomedical, a company that does worm aging screens. So we'll be talking more about drug screening in that episode. And I want to leave you with a final video on a weightlifting collaboration of the century between Arnold Schwarzenegger and Ronnie Coleman.
Where it goes, Jim Venice, and I'm about to get me a workout in with my boy Arnold. Let's do this. Yeah, buddy. Want to go too heavy here?
No, God forbid.
Yeah.
For retired people like us,
Nope.
you got to take it easy.
Exactly.
Exactly. That's enough, Ronnie. You're exploding. Stop it.
Come on, lightweight. Let's go. There you go. Show them how these old men work out.
I
Lightweight.
can't. Is anyone older around?
Right here.
We have a combined 15 Olympias and combined 125 years
Old.
age. Don't wipe it off, Ronnie. I want to use your sweat to get some power here. Look at those bumps sticking out here.
Oh, man.
Oh, look at that. You see, that's the difference between this 8-time Mr. Olympia and
And a 7-time?
and the mere 7-time Mr. Olympia.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.